Bismillahirrahmanirrahim. The physiological function of growth hormone are discussed in this video. All the hormones from the pituitary gland, they affect on target glands such as the TSH effect on thyroid gland. Some hormone go to the gonads while some on the breast or other organs of the body or target tissue of the body. But the growth hormone is unique in nature because it affects or it exerts its effect on whole of the body. So this can be visualized as an analogy picture. This pituitary hormone is a control tower, and at the airport where each hormone is a flight controller sending instructions to different planes, their target glands. The thyrotropin controller signals to the thyroid jet, the corticotropin controller guides the adrenal aircraft, and the gonadotropin directs the ovaries and testes plane. And prolactin lands at the milk terminal, that is the mammary gland. But growth hormone is different. It's the maintenance crew working directly on every part of the airport, all tissues, repairing, building, and upgrading non-stop without needing to radio a specific gate or airline. The growth hormone is also named as somatotropic hormone or somatotropin. The molecular characteristic of this is that this is a peptide hormone, protein hormone. It contains about 191 amino acid in a single chain, and its molecular weight is 22,005. The general function of the growth hormone, its primary function is the stimulation of growth of almost all tissues that are capable of growth, and they are responsible for this growth by increasing the cell size. That means is promote hypertrophy, and by the stimulation of the cell mitosis. It also uh, causes the cell differentiation, particularly in bone and muscle precursor cell. So here this graph is showing that the growth rate among two determinate rats, and here you can see the one is treated or injected daily with growth hormone, and another rat is a control and untreated. So growth hormone treated rat shows significant increase in growth. The growth effect were these effects were observed during early development and adulthood, and early development, the organs of the treated rat increase proportionally in size, and adulthood, most of the bone is stopped lengthening, but many of the soft tissue continue to grow in post-adulthood rat. The bone length did not increase due to epiphyseal closure. See, so this is a systemic growth promoting hormone uniquely affecting nearly all body tissues directly, unlike other anti-pituitary hormone that acts through target glands. The growth hormone has multiple metabolic effects beyond promoting tissue growth, impacting protein, fat, and carbohydrate metabolism. It promotes anabolic effects on protein and catabolic effect on fat and glucose sparing effect, shifting the body's metabolism toward protein preservation and fat utilization. The protein metabolism is affected by increased protein synthesis in most body cells, increased uptake of amino acid, and decreased protein breakdown that promotes the positive nitrogen balance, and it will lead to increased lean body mass. Then it also affects fat or lipid metabolism. There is increased lipolysis or breakdown of the fat in adipose tissue, and increased release of free fatty acid into the bloodstream. There is increased fatty acid oxidation, and these fatty acids are used as energy source. The net result is the reduction of the fat store that provide alternate energy to the uh, glucose. And then it also affects the carbohydrate metabolism. There is decreased glucose uptake in the tissue and decreased glucose utilization. So there will be increased blood glucose level. So these growth hormones are also termed as diabetogenic hormone. That the net effect on these carbohydrate metabolism, that it conserve carbohydrate, favor fat as primary energy source. So growth hormone is both anabolic and anti-catabolic. Anabolic means it is building in nature, and it is also anti-catabolic, that is preserving in nature, and making it powerful promoter of protein deposition and tissue growth. The growth hormone enhances protein accumulation in the tissue through several coordinated mechanisms that increase the amino acid availability, stimulate protein synthesis, and reduce the breakdown of the protein. So, what are the mechanisms of protein deposition? One is the enhancement of amino acid transport into the cell. So growth hormone directly increase amino acid uptake across the cell membrane, leading to increased intracellular protein concentration or amino acid concentration that promote the synthesis of the protein. So like insulin promote glucose uptake, this growth hormone, it promotes amino acid uptake. Increased RNA translation at the ribosome is also an effect of growth hormone. Here, the growth hormone stimulates the translation of existing messenger RNA into protein even without more amino acid, and enhances cytoplasmic ribosomal activity, increasing the protein output. Then increased DNA transcription in the nucleus is another effect by growth hormone. Over 24 to 48 hours, growth hormone boosts transcription of DNA into RNA, resulting in more RNA available, messenger RNA available for translation that lead to sustained protein synthesis. This is likely growth hormone's most important long-term control effect. Assuming adequate nutrients. Then decreased protein and amino acid catabolism is another effect of the growth hormone. The growth hormone inhibits the breakdown of the protein in this cell. So this mechanism is through the uh, protein sparing effect or of the fat mobilization. So it increases the fat mobilization. So fatty acid replaces protein as an energy source. So this protein sparing effect conserves protein of the body. So in nutshell, The effects are increase amino acid uptake, so increase protein synthesis and decrease breakdown of the protein. So anabolic action as well as anti-catabolic or protein preserving action. So to summarize the protein effect, there is increased amino acid transport that results in increased substrate for protein synthesis. As a result, increased RNA translation occurs and increased protection of the protein occurs at ribosome. There is increased DNA transcription also. So increased RNA and increased protein synthesis occurs. That is a long-term effect of this hormone. Then the protein are preserved also. Their breakdown is decreased. That conserves structural and functional proteins. Growth hormone plays a critical role in mobilizing fat for energy that favors the lipid oxidation over carbohydrate and protein synthesis. The increased release of fatty acid from adipose tissue will result in increased level of free fatty acids and then free fatty acid oxidation in the tissue, uh, and these fatty acids are converted into acetyl CoA for energy. So the energy preference shifts. Now the body uses fat instead of glucose or protein. The protein synthesis begins within minutes of the growth hormone stimulation, and the mobilization of the fat, it takes several hours to become significant, and due to this effect the growth hormone produces early anabolic effect followed by the long-term fat utilization. So the cumulative effect on the body composition is fat burning and protein synthesis that leads to increased uh, lean body mass. The growth hormone promotes muscle building while reduce the fat. The ketogenic effect of excess growth hormone, what is that? The excess growth hormone will result in excess fat mobilization, and due to this excess fat mobilization there is formation of excess acetyl CoA, and this acetyl CoA in the liver, it is converted into acidoacetic acid that lead to ketosis. So risk of fatty liver is due to overload of lipid in liver tissue. So to summarize this fat utilization, the mechanism for mobilization of fat, there is a stimulation of the lipolysis in adipose tissue that results in increased free fatty acid, and then what boosts fat use? Growth hormone boosts fat use because it enhances acetyl CoA production from free fatty acid that causes the increased fat oxidation. Then growth hormone also results in protein sparing. It has got a protein sparing effect. So fat replaces protein for energy and conserve muscle mass. Then growth hormone also causes excessive fat breakdown So too many ketones are produced or ketosis or fatty liver may result. Another effect of the growth hormone is to decrease the carbohydrate utilization. So there will be decreased glucose uptake in the tissue such as skeletal muscle and fat. There will be increased glucose production by the liver, and there will be increased insulin secretion. So all these lead to insulin resistance that make this growth hormone as diabetogenic. And these effects are similar to the effect that are produced in type 2 diabetes mellitus, that is non-insulin dependent diabetes. So mechanism of these is unknown. However, one hypothesis is that the growth hormone causes the increased fatty acid concentration in the blood that lead to impaired insulin action of the insulin, impaired insulin action on the tissue. So increased fatty acid concentration also reduces the sensitivity of the liver and skeletal tissue that alters the insulin effect on carbohydrate metabolism. So the growth hormone reduces glucose utilization primarily by inducing insulin resistance, making it a diabetogenic hormone when present in excess. The carbohydrate utilization can be uh, summarized as the growth hormone decreases glucose uptake in muscle and fat tissue that causes increased blood glucose. Then there is also increased production of the glucose by the liver tissue that also adds to increased blood glucose. There is increased secretion of the insulin that is as compensation for insulin resistance, and there will be increased free fatty acid that worsen the sensitivity of the insulin. So all these are diabetogenic effect of the growth hormone, like diabetes type 2, that is non-insulin dependent diabetes. Remember, the growth hormone reduces glucose utilization primarily by inducing insulin resistance, making it diabetogenic hormone when present in excess. Growth hormone is responsible for growth, and without the pancreas and without the carbohydrate, this effect is not achieved. So adequate insulin and carbohydrate availability are essential for the effect that promote the energy and the amino acid transport. So this growth hormone cannot promote growth effectively without insulin and carbohydrate. That highlights a vital metabolic partnership that is necessary for tissue development and anabolism. Insulin is not just a glucose regulator, it is critical growth cofactor, especially in protein synthesis. So the insulin stimulates glucose uptake, and this glucose uptake supplies necessary energy for anabolic process. It also enhances amino acid uptake that provides building block for protein synthesis and it complements the growth hormone action, enabling the synergistic tissue growth. While the carbohydrates, they are the main energy sources, support energy intensive protein tissue synthesis. Then they also prevent excessive fat or protein catabolism to maintain the anabolic state. So insulin and carbohydrate are essential for unlocking the full anabolic and growth promoting potential of the growth hormone by fueling metabolism and supporting amino acid uptake and protein synthesis. As this growth hormone is responsible for new protein formation and growth, it is also responsible for the growth of skeletal system, especially it stimulates the cartilage and bone growth. So there are several mechanisms. There is deposition of protein by the chondrocyte and osteogenic cells, and then there is increased rate of reproduction of these cells. Say these cells are produced more, divided more, then there is a specific effect of converting the chondrocyte into osteogenic cell, and all these effect will lead to the increased bone growth, and the growth skeletal frame increases. Growth of skeletal frame is increases as a result of these multiple factors. So bone increases both in length, and also there is osteoblastic activity, increased osteoblastic activity. So before the uh, closure of the epiphysis, these effect, the growth of the growth hormone effect, there they will cause deposition of new cartilage, conversion of the cartilage into new bone, then elongation, and also responsible for the pushing apart of the epiphysis that result in increasing the height of the person. So there is also increased osteoblastic activity near the periosteum and some bone cavities, and there is also deposition of the new bone, and as we know that these osteoblasts are responsible to deposit new bone, and they also responsible for removal of old bone, because they work in cohesion or they work in concert with the osteoclast. So there is removal and simultaneous deposition of the new bone. So this deposition is more as compared to the removal of the old bone. Then also there is uh, increase deposition, especially in membranous bone. So that will increase the uh, thickness of the bone and results in various protrusions such as occur after adolescence. This protrusion of the jaw occurs, or protrusion of the frontal uh, prominence of the skull. All these are result of these growth hormone effect. So growth hormone plays a critical role in the skeletal development, especially during childhood and adolescence. It stimulates both the long bone lengthening and thickening, influencing the height and bone structure. So the effect on the skeletal tissue is it affects the chondrocyte, osteogenic cells, and osteoblasts all. There is increased protein synthesis and increased cell production of the chondrocyte. Osteogenic cells stimulate conversion from chondrocyte, so they are transformed into osteogenic. cells. Then osteoblasts are responsible for increased bone deposition and increased thickness of the bone. There are two mechanisms of the bone growth. One is the longitudinal growth that is responsible for lengthening of the long bone. So the epiphyseal cartilage is stimulated by the growth hormone because there is cartilage deposition, and then it is later replaced by the bone. Then bone shaft elongates and push the epiphyses apart, that results in increasing the or lengthening of the bone. So the end point is that epiphyseal plate closes uh, in late adolescence, so there is no more growth. The second pattern or second uh, growth pattern is the thickening of the bone or appositional growth. Here the osteoblastic activity deposits new bone on outer surfaces, and osteoclastic activity removes old bone from inner layer, like a seam effect that is described in playlist of the bone and skeletal muscle disorder. Then the net effect of these is increased bone diameter. When deposition increases, resorption of the bone. So the net effect of the growth hormone on the body skeletal system is that during the pre-adult life there is increase of the bone length. Because this growth hormone increases chondrocyte and osteoblast activity, and cartilage and bone both are affected, that results in lengthening of the bone. And when there is increased osteoblast activity, that is more than the lytic activity by the osteoclast, so there is thickening of the bone, and this process occurs lifelong throughout the life. So chondrocytes, they also transform into osteogenic cell. So the cartilage may form bone. So the bone formation accelerates. So the growth hormone drives long bone lengthening during youth and bone thickening throughout life, shaping the skeleton through its influence on cartilage, osteoblast, and overall bone metabolism. When the growth hormone is applied to cell culture, there is no effect produced by this growth hormone on the cell culture. So when while it has got growth effect in vivo. So it is postulated that liver mainly and some other tissues are responsible for formation of somatomatins. These somatomatins are responsible for most of these effect of the growth hormone. Somatomatins are also termed as insulin-like growth factor, and there are four types of this somatomatins, and most common is type C. The molecular weight is about 7,500, and its concentration follows the concentration of the growth hormone. So these somatomatins are produced by the liver, and they mediate the many of the functions of the growth hormone. And somatomatin C is also termed as insulin-like growth factor 1. Pygmies of Africa and Levi's dwarf, they are not able to form these somatomatins, and due to deficiency of somatomatins, these conditions these are represented with dwarfism. However, when growth hormone is injected into intraepiphysis, in the epiphysis there is cartilage growth, and it is postulated that there is local production of somatomedins by the within the epiphysis that is responsible for local action of the growth hormone. Somatomedins or insulin-like growth factor 1 or type C somatomedin, they act as essential mediator of growth hormone effect on growth, especially bone and cartilage, though growth hormone may also have direct local effect in some tissue. So this growth hormone stimulates liver tissue to produce somatomedin. Somatomedin mediate growth hormone effect, especially skeletal effect, and growth hormone may act directly in some tissue. Causes of the dwarfism is somatomedin deficiency. Thank you for watching this video. If you have any query, please write in the comment box.